Okay, so good afternoon, everyone. Welcome to this session, which promises to be very exciting. And it's a pleasure for me to be here as a moderator of the session. So my name is Delphine Croisier, and I'm the founder of Vivexia. So we operate in the field of preclinical evaluation of anti-infectives, and we are paying a particular attention to the humanization of pharmacokinetics in our models. So today we are gonna hear for a panel of brilliant minds who are working hard to contribute to the AMR efforts and to come up with concrete solutions to deal with high priority pathogens and or persistent infections. So our speakers will have time for 10 minutes of presentation and about 4 or 5 minutes for Q&A. And as I'm convinced that this session will raise many questions, I see many experts in the room, so feel free to join the speakers after the session if you want to continue the discussion. So with that, I would like to invite our first speaker. So it's Dr. Christophe van Emmelen. So Christophe is the CEO of Orbilytics, which is a company based in Belgium. So one could say that Christophe is the true serial entrepreneur, as he brings over 25 years of experience in drug discovery and executive leaderships across multiple therapeutic areas, and among them infectious disease. So, Christophe, the floor is yours.
Thank you for the very kind introduction, Delphine, and thank you all for joining the session. I'm genuinely excited to be here and to have the opportunity to present the vision behind Orbilytics. And that vision is to make a meaningful impact for patients worldwide whose health and unfortunately also whose lives are at stake as a result of untreatable bacterial infections. And as Mark and I were chatting just before the meeting, without diversification, AMR will continue to outpace innovation. And like in cancer therapy, where biologics, cell therapies, RNAi drugs have not only unlocked new targets, but also significantly improved patient outcomes, multimodal arsenal is equally essential to provide sustained control of bacterial infection. And so it's within that context that we at Orbilytics are developing precision licensed therapeutics. It's our version of engineered license, PLTs as we call them. And we developed them as a novel modality capable of very potent and selective killing of pathogens, including gram-negative pathogens, through a unique mechanism of action that does not drive antimicrobial resistance. The breakthrough that we bring to the field is that we've been successful in overcoming a technical restriction that for decades has limited the clinical utility of engineered license specifically against gram-negatives. And we've been able to unlock the therapeutic potential for how to treat hospital-acquired infections thanks to a proprietary platform that not only allows to overcome the technical hurdle, but also to scale and to allow a build-out of a pipeline targeting multiple gram-negative SKP pathogens. Today, I would like to illustrate, firstly, introduce the modality as such, illustrate how that platform has allowed us to identify and optimize PLTs with potent bactericidal activity against gram-negative, but most importantly, maintaining that activity across a range of physiological matrices, and how that in vitro potency compellingly translates into meaningful in vivo efficacy. I will also show the broad spectrum activity across a panel of bacterial strains before concluding with data that actually support the mechanism of action being enzymatic lytic degradation of the peptidoglycan layer, underpinning the therapeutic potential of this modality. So PLTs, or precision licensed therapeutics, unlike conventional antibiotics, act via direct and targeted degradation of the peptidoglycan layer in the bacterial cell wall. Through this non-metabolic mechanism of action, they're not only capable of very fast, irreversible bactericidal activity, but they also inherently limit the number of routes to which resistance actually can emerge. As you can see on this illustration, each PLT is an engineered multi-domain protein containing various functional activity domains that all work in a concerted manner. There is the outer membrane peptide, which is responsible for translocation across the outer membrane, which is so characteristic for gram-negatives. There's a cell wall binding domain that adds both specificity as well as docks the PLT on the bacterial cell wall. And last but certainly not least, there's the enzymatic activity domain, originally derived from endolysins produced by bacteriophages, capable of very fast enzymatic degradation of defined peptidoglycan bonds. So it's this disconcerting mechanism that is actually capable of very fast killing, literally within seconds to minutes. The breakthrough that we as a company have introduced in the field is, as I mentioned before, we succeeded in designing and engineering this lysins to remain fully killing under high serum concentrations. So what makes PLT so uniquely compelling is that they not only have these inherent features, but they are also one of the very few, if not the only modality, that also meets all the innovation criteria as imposed by the WHO. So in essence, this is not just an incremental expansion of existing antibiotic classes. For the first time in decades, we have the opportunity to truly transform how we treat bacterial infections with a modality that combines speed, precision, and resistance resilience that conventional antibiotics simply cannot match. This progress has been powered by a proprietary platform that integrates synthetic biology, bioinformatics, and automated screening, importantly, straight away under physiological conditions. The real power, however, in our approach resides in the fact that we are capable of generating very massive, multidimensional proprietary datasets that we can mine, that we can subject to machine learning to unveil design principles, but we can also apply in AI-enabled prediction, allowing us to significantly shorten the time for library design, but also for hit-to-lead lead optimization iterations. And all this actually turns PLT discovery into a precision-driven, scalable process. This platform has allowed us not only to sample a very vast theoretical design space in a fast and efficient manner, it has also allowed us to identify those pair variants capable of maintaining potent bactericidal killing under high serum concentrations. So what you see here is the comparison between PLT2, which is a non-optimized lead identified through screening, versus CF370, originally discovered by Contrafex, and often used as a benchmark for engineered lysins against gram-negatives. The data actually come from a CFU reduction assay, basically a single type 1 killing assay, and as you can appreciate by the height of the bars, PLT2 in pink is capable of maintaining very potent bactericidal activity across the full spectrum of human serum, all the way up to 80%, so truly mimicking real-world systemic circulation. These results are in stark contrast with regard to CF370. As you can appreciate, CF370, despite the fact that it has a potent bactericidal activity at 0% human serum, completely fails once it's being exposed to even 10% human serum. So these breakthrough data not only validate the approach, they also served as a launchpad for a subsequent lead optimization program, allowing us to design and engineer even more potent PLTs. What is important is that this potent bactericidal killing activity in vitro very convincingly translates into meaningful in vivo efficacy. In these results from a mouse Acinetobacter baumannii bacteremia model, you can appreciate that PLT2 is capable of efficacy comparable to tigecycline, a commonly used antibiotic for the treatment of multi-drug-resistant gram-negative infections. So after intravenous dosing of PLT2 at 2 and 4 hours post-infection, using a clinically relevant dose of 10 mpk, PLT2 was capable of statistically significant reduction of bacterial burden in a number of major organs, including blood, as you can see depicted here. But more importantly and more impressively, and this is the blue bar, when we treated the mice with tigecycline and on top we added PLT2, what we saw and observed was statistically significant in vivo synergistic efficacy. So these in vivo data truly validated the therapeutic potential of PLTs, not only as a standalone therapy, but more importantly and clinically more relevant, also as a potential alternative for adjunct therapy on top of standard of care. It is important to confirm that this potent activity is not just restricted to a lab strain, that this equally applies to clinically relevant strains, and particular strains that actually have been isolated in the clinic and have been known to have certain resistance mechanisms specifically against standard of care treatment in a clinical context. So what you see here is a head-to-head comparison between PLT2 and colistin, using a panel of strains, some of which have resistant elements against colistin. You will appreciate by looking at the MIC values that whereas some of these strains lose all susceptibility to colistin treatment, they remain fully susceptible to PLT treatment, with MIC values that are predominantly in the submicromolar range. So these data and other data against other clinical isolates really demonstrate the clinical relevance of a PLT, whereas physicians often rely on adjunct therapies like polymyxins or tetracyclines. The resistance is rising against these agents, as well as the clinical use being restricted by toxicity. So with these data, we believe that we can actually offer an alternative adjunct therapy by using PLTs on top of standard of care. Before concluding, I wanted to show some data that really confirm unequivocally that the killing activity is really driven by the enzymatic activity domain. So here what you see in the left panel is basically a rendered model of PLT2 with a peptidoglycan substrate docked in the catalytic site. The top panel in the middle basically shows the results from a biochemical assay where we looked at substrate turnover, with the red curve at the bottom depicting the very nice gradual turnover by PLT2, the parental engineered lysine, as opposed to the superimposed dotted lines indicative of complete loss of activity following the removal of essential amino acids in the catalytic site. What is important is that these catalytically dead mutants translate very nicely the loss of enzymatic activity in the loss of functional activity, as is shown in a panel in the bottom middle. There you see a very nice and a very compelling shift in potency in terms of their killing activity, as is also shown in the table on the right with the corresponding EC50 data. So what these data actually confirm is that, yes, it is enzymatically driven peptidoglycan degradation that drives the bactericidal activity, and it's indeed this type of orthogonal mechanism that also underlies the high resistant resilience that is so characteristic for this type of agents. So before concluding, I'd like to draw your attention to these two images on the right. These are scanning electron microscopy images that were made by treating an Acinetobacter baumannii bacteria with a PLT for only 5 minutes. You will appreciate that even after such a short period of time, the PLT is capable of instantaneous irreversible perforation of the bacterial cell wall, which triggers massive expulsion of cytoplasmic content and immediately triggers a full structural collapse, leaving behind nothing but bacterial debris, which is shown in the panel on the right. So with these data, I hope I've been able to convince you and get you excited about the transformational potential that PLTs have to offer. We are currently advancing our beachhead project for multidrug-resistant Acinetobacter baumannii BSIs, going forward towards development candidate nomination, and at the same time are expanding our pipeline into other additional high acuity indications triggered by gram-negative SKB pathogens. With this, I would like to thank the team. I'm only the messenger. You see here all the people who have been responsible for all these results. I would also like to thank our partners at the UK Health and Security Agency, our financial backers, and you for your kind attention. I would be happy to take any questions.
Many thanks, Christoph. So if some questions arise in the audience. One question before that. I have one for you. So you mentioned the lead indication would be the sepsis, am I right?
Not sepsis, bloodstream infection.
Yeah, bloodstream infection. But considering the main entry is usually pneumonia in the nosocomial settings, do you plan to develop this compound in the pneumonia context, considering that higher inoculum can be found in this diseases?
It's a highly relevant corporate strategy type of question, and we gave it a very, very long and hard thought. Eventually, we decided to prioritize BSI over HAPFAP for a number of reasons. In terms of clinical feasibility, the number of endpoints that you can actually use to demonstrate clinical proof of concept are a little bit more amenable to the type of modality that we're using. But equally, we believe that despite the fact that BSI is a more focused, accessible market, the fact that we can not only go for fast clinical proof of concept, but also rapid market entry, allows us, based on the unmet medical need, to also go after premium pricing. And from there on, obviously, we intend to extend into other strategic indications, and multidrug-resistant Acinetobacter HAPFAP would be the next one.
I think, that's a great the study. So I think it's really the issue to have the serum stability in gram-negative, right? But my question is, what structural features drive serum resistance in your PLT?
I think I probably may discriminate two parts in your question. So one is structure, property, function, and the other one is resistance. We're not in a position yet to disclose the data. We hope to be able to disclose them really, really soon. But so far, everything seems to point to an extremely high hurdle for resistance emergence. So I hinted at that when I emphasized the orthogonal mechanism of action. We're currently concluding a series of experiments where we, data-wise, hope to substantiate that claim, and we hope to be able to share that really, really soon. The first part of your question is with regard to structure, sequence, structure function. That's very much the core of the platform that we operate. That is really the basis of how we basically analyze the data, try to unveil design principles, and apply these accordingly. We use both human interpretation as well as machine learning, where we look at features that the human brain simply cannot understand. And so these uninterpretable features are largely included in the AI-enabled prediction tools.
So for the clinical indication, I understand why you're going bloodstream. Are you, for trial design, are you thinking about doing with co-administration of an antibiotic? Because I find it hard to believe a doctor would do this on its own.
Absolutely.
And contrainfected that as well. Okay, yeah. So,
No, absolutely.
so then the follow-up question to that is it, I would think it would be interesting if there's certain synergy with certain antibiotics that you might have with one versus another. And have you explored any of that so far?
So the reason why tetracycline was used in this particular model is that's what the model was validated against. Indeed, to your point, we need to test this also in combination with other standard of care. In vitro, we've already done so. The next step is obviously to also corroborate that in vivo.
And how many classes in vitro have you tested so far? Like all 26 classes of antibiotics?
No. Okay. Work in progress, but very promising data.
Great talk. Since the license degraded cell wall, did you find LPS release?
Deep.
LPS release?
It's something that we are aware of. Certainly, once we go into the clinic, we have to be mindful that if we should have any massive lytic event releasing, indeed, components like LPS, that we not have some sort of a pyogenic reaction. So that's definitely something to monitor very, very carefully. That's also where careful PK/PD modeling will be absolutely essential.
Yeah, last question.
Just a question regarding the slide 9 on the PMRB mutant. There is a significant MIC difference between the wild type and the PMRB mutant. So I'm just guessing that if you treat a patient that has been treated with colistin and you cannot cure the infection, the PMRB is also involved in the change of the LPS on the surface. So it might be that the lysine will be less effective against these bacteria in this specific patient. So I'm just guessing, would you find a way to overcome this?
Firstly, the data that were shown are the data from our most advanced frontrunner, which is still a contender as a potential development candidate, but not necessarily so. So we're also advancing other leads and characterizing these extensively. I would also think that we have to be mindful of overinterpreting these data in the sense of what's the error of margin. So I would argue that more exhaustive testing is required as well.
Thank you, Christophe. Many thanks again.
Thank you.
So I would like to move to our second speaker, Shubhendu. Is it right? Basu. So Dr. Shubhendu Basu is the CEO of Telum Therapeutics, and with over 2 decades of leadership experience in biotech and pharma, he brings expertise in immunology, infectious disease, organizational growth, product innovation, so and so. And before joining Telum, he served as president and CEO at Adaptive Phage Therapeutics. So, Shubhendu,
Thank you,
yours.
Delphine. Yeah, very kind words. It's intimidating to be in front of such a large audience. Can you hear me okay? Good? Okay, perfect. So I like to look at the slides when I speak, even though they're up there. So Telem Therapeutics, we're based in Pamplona, Spain. That's where we're headquartered. And essentially what we're doing is we're going straight from metagenomes to precision antibacterial therapeutics. Metagenomes, which are proprietary, we mine them, and then we get straight to our license-based therapies. So I'm not going to dwell on the problem of AMR. We're all here for that person. It needs to be addressed. That's why we're here. There's a lot of death, morbidity, mortality. But, we're still struggling. AMR is still an issue. And the question is why? Why is it still an issue? And we're not going to be able to solve the problem by repeating what we've been doing for the past 50 years. It's not going to work. What happens is our drug development cycle is slower than evolution and the rate of biology. So we have a broken model for antibiotic development. Investment is fallow. We all know that. No one really invests in this anymore. Resistance emerges with use rapidly. Oftentimes there's resistant strains that exist even before a drug comes to the market, an antibiotic, that's just selected for with the use of the antibiotic, which leads to limited clinical durability. It drives stewardship. Stewardship is excellent. However, if you're the innovator company that spent millions of dollars getting a drug to market and no one's using it, you're in trouble. So we've gone through this whole process of people getting pushed across the line and still going bankrupt and not having any return on their product. So what does Telem do? We are fundamentally trying to change this process, right? So we have what we call engineered phage lytic enzymes. It's probably a different variant of a PLT. And it's a platform-based system. The key is we leverage proprietary metagenomes. So we don't mine public databases because we are critically aware of IP, FTO, and we're running into those issues. So everything that we have is not in the public databases. It's driven from proprietary metagenomes. We have a continuous learning loop. I know AI is a big word. I'd like to say AI, but I think what we really do is machine learning. AI is something I don't fully comprehend yet, and it changes every second, but we're definitely driving it based on machine learning. It's experiment, fed back to data, fed back to pivoting. And so what do? So we have a proprietary metagenomes, and then we have license. But we don't take national license. We don't take the license that are there. We deconstruct them. We look at individual domains of these license. And then we test each one experimentally for the desired effect we want. And then we have these engines that recombine. So we deconstruct and reconstruct, right? These licenses to make unique licenses that then go through this whole list of aspects that they have to clear, be it manufacturability, stability, activity, enzymatic activity, so on and so forth. And at the end of it, all of that goes back to the learning loop that then gives us the next lead. So this is some quick data. I think you may have seen this before because we've been presenting this. So our lead asset. So we have assets for staph, Pseudomonas, Kleb, E. coli. But the lead asset we have, which we call TTX1, is actually a broad-spectrum gram-negative. It's active against Ebomania, Pseudomonas aeruginosa, E. coli, and Kleb. And you see the MIC values in there and the frequency with which these isolates are there. Now, this panel is nice, but our goal is to expand it to more than 100 isolates or 200 isolates for each of these bacteria. So the one thing over here about the durability that I was talking about is the emergence of resistance. So the graph over here, oh yeah, there we go. So the graph over here, what this shows you is, so this is cefiderocol, meropenem, and colistin. Can you hear me okay? Yeah. So essentially what happens is we take these bacteria, put them into subinhibitory concentrations, and do serial passages and look for the emergence of resistance. And this is primarily what we've been fighting all these years, is that within a few passages, you see resistance emerging. Whereas with our lead asset, you don't see any resistance emerging to Ebomania, Pseudomonas, or E. coli. So this goes to the durability, not just the effect at a point of time, but over time. And that's one of the key aspects we look for before we advance something. So, Christoph alluded to contraeffect and stability and all of that. So I'm glad to say, so yes, serum stability is important. We test our proteins in serum too. But we've noticed that to have the same level of activity, the actual amount of protein you need to put in has to go up. So you can't have the same concentration or same quantity of protein and get the same effect. So you can get serum stability or serum activity, but with increasing amounts of protein. So using that proxy, what we then decided to do is, okay, how does it work in animals, right? So in vitro is fine, but how does it actually work in animals? Some of the things you'll see historically is there was slow infusion of these lysins. Repeat dosing was not conducting. ADA is a question, and toxicity. And does it actually kill the bacteria and eliminate them as opposed to maintaining it, right? So we have data that addresses all of this, but I'll just focus on one slide here. So the panel on over here, this, here we go, sorry about that. So this is basically radiolabeled protein, tail vein injected, IV. We look for biodistribution. And you can see that, and this study was conducted for 6 hours total, because that's when they stopped it and sacked them. But you can see that the protein is in the lung within 15 minutes, and it stays in the lung for at least 6 hours at the point it was stopped. It also goes to the liver, and it goes to the kidneys. So the protein distributes when it's given IV to the site of infection. So now the question is, okay, it gets there. Does it work there, right? Is it still effective? So you see the panel on the right is, so this represents 3 independent experiments in mice where there's a lethal AMR lung challenge model. And essentially, all the mice that were not treated, right here, all the mice that were not treated die. Colistin gives you about 70% protection. And our protein at the optimal dose gives you 100% protection. So this represents at least 15 mice, repeated, and you can see 100% protection with protein alone. And this is the colony counts, right? So you see this is in the control, this is the baseline. So you see there's a productive infection because it goes up in the controls and there's mortality. Colistin reduces it as slightly below baseline, and TTX1 has a significant log reduction of CFUs in the lung, and even more so in the blood, right? 5 log drop and a 4 log drop. And this was not one day. This was done over 3 days, right? So you can see it's a 72-hour treatment. So what this study lets us show is we can IV dose in a bolus, repeat dose, and confer protection, and actually reduce bacterial colony counts. So the market, everyone talks about the market, how large is it? Our goal is to enter the market initially with HapWap Abomanii, and the reason we're doing just one bug, even though, like I showed you, it's active against Pseudomonas, E. coli, Kleb, we're going to go in with Abomanii HapWap because there's a high unmet clinical need. Once we have that figured out, we'll expand into adjacent indications with Abomanii, and then other gram negatives with what comes into the platform. So this number there, 2.5 billion, you know. So this was done by an independent market research group. This is not our numbers. But I don't believe that number. That would be a great number to have. I think anywhere in the 400, 500 million range would be fantastic. So historically, what we've seen is past approaches have used a single asset, or they have used public databases, or in silico optimization. Essentially what we're doing is it's a platform-based discovery. We have our own proprietary metagenomes. FTO is great. Refractory to resistance, as I demonstrated previously, and it's compatible with IV dosing and repeated systemic use. This is where we are now. TTX1 is a lead. We have a couple of candidates right behind it. That's the pipeline. So we've also met with the FDA and European regulators. And actually, we had a very positive meeting with the US FDA, and we even have QIDP designation for a lead asset. So they've reviewed their data set, they've granted us QIDP designation, and we have positive feedback from EMA. So why am I here? I'm trying to raise money, not just here, everywhere. And, it's been a slog, but we continue to try to raise money to get this asset into the clinic. So that's where we are. This is the team that actually, the brains behind all of it. So there's Roberto, he's actually here. He's the co-founder and COO, Diana, Irene, and then that's a board who guides us and keeps us on the straight and narrow. And that's us in a nutshell. Thank you.
Many thanks, Shubhendu, for this nice presentation. Maybe there are some questions in the audience. Yeah, Amélie?
You've shown some MIC panels for your broad-spectrum compound with some high MIC pseudomonas in particular. Have you identified the reason for those resistance? It is linked to the outer membrane structure?
So honestly, I don't think we've picked each of those isolates and done a deep dive to understand why they didn't work. We were looking for, it's actually the other way around. We're looking for an asset that has broader coverage. So we would probably just flush them. And we have other assets for Pseudomonas that have better coverage, which is why this lead asset is going in for A. baumannii, but we have other Pseudomonas assets behind it. But once again, what the board tells us to do is stay focused. Because if we leave it to the scientists, we will be running down assets for every bacteria and looking at MIC. So we just need to pick one and run with it, which is A. baumannii for now.
Any other question for Shubhendu? Yeah.
Very impressive in vivo data, Shubhendu, and congrats. Could you maybe comment on the actual dosing regime that was used for this study?
So it was IV, and it was once a day, I believe. Once a day, Roberto, or twice a day? Once a day. Once a day. IV, tail vein injection, once a day, over 3 days.
And the dose levels?
The dose we did from 5 micrograms, we did toxicity at 10 micrograms. We still have to go lower. The problem we have, so I didn't show another study we have where we see synergy with colistin. But over there, the drug was administered IP. So, the drug availability intraperitoneally is not as much as intravenously. So what we actually have to do is go down. We have to start going left because we're getting too much activity at the dose we're going in. So we have to start going left to see where we can get partial survival and then the synergy.
Very nice, thank you.
Yeah.
Thank you, Shubhendu. Many thanks.
Thank you.
So we will move to third speaker. Yeah. So Dr. Yun Yen, am I right? Okay, thank you. Who is the director and R&D executive of the R&BD Institute at Incheon Biotechnology, which is a South Korean company. So Dr. Kim leads programs developing novel antibiotics, anti-cancer agents, and immunotherapy. So before that, he was a co-founder, CTO, and CSO of LifeHome. So the floor is yours.
Thank you for a nice introduction. It is my pleasure to introduce today's talk at the cutting edge session of Vital AMR conference. So my name is Young Kim, as I introduced, and I'm from Incheon Biotechnology. Today, I would like to introduce novel endolysin therapeutic, specifically targets of deadly infection of infective endocarditis. To address this challenge, we developed SL200, as known as Tabana case. So let me briefly introduce our company. Incheon Biotechnology is dedicated to development of innovative drug development using 2 platform technology. The one is Itrysin platform, which is endolysin engineering platform. Another one is IMPA platform, which is specifically functional fuzzy engineering platform. Together with these platform technology, we expand our pipeline from infectious disease and the microbiome-based oncology, vaccine, and immunotherapy. In fact, SL200 was generated from Itrysin platform technology, and also we are conducted 2 gram-negative bacteria using Itrysin platform technology as well. So let me focus on SL200 from now on. So main target indication of SL200 is infective endocarditis. Please look at the left side of illustration of heart. This is pathology of infective endocarditis. Please look at the red circle. The massive bacteria was infiltrated and forming vegetation on the heart valves. The vegetation can protect, allowing proliferation of bacteria within the shield of environment. As a result, infective endocarditis remains serious life-threatening conditions. Despite we have SOC treatment, in hospital mortality still remained 20 to 40%. So in this case, many of our patients require the open heart surgery and prolonged ICU stay. So this is representative not only the serious clinical burden, but also subsequent economic burden as well. The major causative of the pathogen is Staphylococcus aureus, including MRSA and MSSA. So here we have a question: why still remains mortality still so high, even though we have standard of care? Please look at this table. So there are standard of care such as vancomycin and daptomycin. These two or more standard of care faces the three fundamental barriers. First, they are slow very late. That's meaning that they are washed out before properly working. Second, they cannot penetrate biofilm and vegetation, therefore inactivity against the dormant cells. The third one is long-time treatment induces the high-dose toxicity, including nephrotoxicity and musculoskeletal. Therefore, to prove this unmet need, we developed SA200. So SL200 is recombinant endolysin derived from bacteriophagy, and very highly specific for Staphylococcus genus. So particularly including MRSA, VRSA, and even other multi-drug resistant strain. Structurally, SL200 has SH3 binding domain precisely binding to the bacterial cell wall, while two catalytic domains such as CHOP and amidase 2 physically cleave to peptide glycan layers. Therefore, we should remember that this SL200 works through enzymatic hydrolysis mechanism of action, unlike to the other conventional antibacterial, such as they are really rely on the bacterial metabolism. So please look at this comparison graph. This is lytic activity between SL200 and the current standard of cure, so daptomycin, at same mass and molar concentration. SL200 induces rapid turbidity reduction at same mass and molar concentration. In the contrast, daptomycin shows no measurable lytic activity at all within the same time frame. So you might be wondering, how about the biofilm issues? The biofilm is one of a serious problem to treatment failure in infective endocarditis. So please look at the left side of the picture. This is before SL200 treatment. SL200 penetrates biofilm and rapidly lyses the embedded bacteria. When SL200 lyses bacteria, lyses the enzyme further destroy remaining biofilm structure itself, indication that SL200 eliminated the persistent bacteria within the shield of vegetation. Okay, let me move to the other story then. What about the antibacterial resistant issue? So we performed that this antibacterial activity against methicillin susceptible and methicillin resistant strain, and vancomycin resistant strain, and the daptomycin resistant, even with the linezolid resistant strain. And then we got the really good no cross resistant with the conventional antibiotics. Then what about in clinical sample? We isolate over 100 clinical samples from patients. So please look at the left side of the table. So out of 100 clinical samples, over 90% of isolate were inhibited at concentration of 4 microgram per ml or less, MIC 50 over 1 and the MIC 90 over 4. And particularly, this range is extremely narrow and consistent potency range. Importantly, we also look at the antibacterial activity in between MRSA and the MSSA strain, and particularly we got a really strong antibacterial activity in both of MSSA and MRSA, indication that no cross-resistant issue at all. And importantly, please look at the right side of the table. 21 out of 29 MRSA clinical isolate a multi-drug resistant profile. So green R means indicates that each individual had a specific conventional antibiotic resistance. And then we got such a good number of MIC value, 1.19, indication SL200 has no cross-resistant issue at all, even with the multi-drug resistance. So then you might be wondering whether SL200 has a synergistic potency. So therefore, we already knew that SOC such as daptomycin. To address this question, we conducted a checkboard dilution assay with daptomycin and obtained FIC index 0.25, which indicates that extremely good value of synergistic potency. So let's move on to the safety issue. Because the GLP toxicology study and the safety pharmacology is extremely enough and important prior to the SL200 administering to humans. To that, we conducted a general toxicity in mice and rat and dog and monkeys, and then obtained no target organ toxicity issues. And also, it is quite important to protein drug-related issue. And then we have very safe profile, and then no severe vascularity and hypersensitivity at all. And then also, we have very clean safety pharmacology issues, and then obtained no adverse effect on central nervous system, and the respiratory and the cardiovascular systems, even human ERG levels. And also, we have a very clear and excellent safety profile from the human trials. We conducted in phase 1A study a single ascending dose, and then phase 2B study multiple ascending dose in human volunteers. As well as we also obtained really good data, phase 2A, patient with persistent SRS cyst bacteremia. In particular, phase 2A add-on therapy, we observed no additional toxicology at all. This is indicating that SL200 is really clinical applicability. Okay, let's move to the rapid PK study. We evaluated how dosing frequency and the fusion duration affect on the systemically exposed of SL200. We optimize dosing frequency as BID or TID, and the infusion time frame from 10 to 60 minutes. The BID meaning that twice SL200 treatment twice a day. TID means SL200 treatment 3 times a day. This is the result of rapid PK study. So as you can see in this table, at 60-minute infusion, maximize therapeutic exposure compared to the short time infusion, compared to the 10 to 20 minutes. Therefore, the subsequent in vivo efficacy study was conducted for 60-minute infusion with BID or TID regimen. Okay, let's move on to the rapid efficacy study. We established the catheter-induced left side infected rabies model. This is really a mimic of human disease, also one of extremely difficult to treatment with the standard of care. The rabbit was infected high virulence MRSA MW2 strain, and SL200 treatment either 27 milligram per kg or 15 milligram per kg in combination with daptomycin, together with a BID or TID regimen for 60-minute infusion for 3 consecutive days. So this is the rapid efficacy result. Please look at the left side of the table. This is such a wonderful data that recently we got. So bacterial burden from the cardiac vegetation and kidney and the spleen show dramatically reduction at the treatment of SL200 at 27 mpk compared to the daptomycin only. Remarkably, in case of treatment 50 mpk of SL200 achieved complete sterilization of all organs, including cardiac vegetation and kidney and spleen. This is also what we saw that bacteria count reduced below the limitation of detection, the meaning that less than 1 log 10 cases. This is extremely important and exciting data. Such a data is never achieved with a conventional SOC. So based on this good result, we also obtained phase 2B IND approval from United States FDA. In this case, we target MRSA or MSSA bacteremia with the right and left side infective endocarditis. Okay, let me summarize my talk. Please look at this table. The conventional antibiotics has very limitations, such as low react, and also they have a high prevalence of multi-drug resistant strain. Also, they cannot penetrate biofilm and also vegetation, as result in also cannot properly working against the dormant cells. Also, they have a really big issue in case using high doses, such a toxicity induced. However, SL200 is a completely different solutions. SL200 has rapidly bactericidal effect, and also we are able to penetrate bacterial film and vegetation. Therefore, we are actively against the dormant cells. In addition to this, we have no issue about cross-resistant issue, even with the multi-drug resistant strain. And importantly, we also conducted that good, excellent safety profile, not only animals, but also humans. Therefore, I would say SL200 is breakthrough medicine to address current standard of care. Let me finalize my talk. We are actively seeking of global partners to licensing out or co-development of late stage of SL200. Meantime, we also interested to the co-development or like a strategic the licensing out, especially target of gram-negative bacteria using our innovative interesting platform technology. Let's shape the future of AMR therapy together with the Interim Bio. Thank you for your attention.
Thank you. And congratulations for the rabbit study, because coming from this field, we know the large amount of work it represents. Yeah. Any question for Dr. Kim? Yeah. The microphone there, just behind you.
Hi, great talk. I have a naive question. I remember seeing SL200 was in the clinic for Staph aureus bacteremia a couple of years back. Is this molecule different from that molecule, or is it the same molecule, but now you've switched indications?
It's the same molecules. The switch is different indications.
Okay, thank you.
Yeah, one last question.
Thank you. What did you learn from contrafact studies? Because I had quite promising study in phase 2, but then failed in phase 3, also going for bacterium, for
the contrafactor, the other name is exacerbacase, also using the endolysin, right? Similar like ours. But main difference contrafactor and ours one is they are fundamentally different of features, because we have two catalytic domain, including CHOP and amidase 2. However, contraphase has one catalytic domain, only CHOP. And also they cannot work MSSA strain, only works MRSA. So if you look at the economic view, so the equivalent, the target market, MRSA and the MSSA, but they cannot work. So I don't know why they consider to progress phase 3, because even they have the phase 2 exactly the same, right? But as I told you, we have a completely different molecule. And most importantly, we don't have any of side toxicity effect at all. In case contrafactor using 1.4 milligram per kg in animal, they induce the vascularity. But even we are using the 50 milligram per kg in animal models, we don't have any of vascularity or hypersensitivity at all. Also, we proved that we have a strong evidence also conducted to phase 1A and phase 1B, and even patient with phase 2A, we don't have any issue of safety profile. This is the main difference and a completely different scenario.
Thank you. I'm pleased to welcome our last speaker, Mark Butts. Oh, sorry.
Oh. No problem.
So Mark Butts is interim CEO of Santero Therapeutics, a Belgian company. So Belgium is in force today. And he holds a bachelor in mathematics. Yeah. with over 25 years of experience in biotech. Mark is what we might call a Swiss Army knife. So, Mark, the floor is yours.
Thank you. I'm not Swiss or Belgian, so that's good. I need the... There we go. I'm the last one, so I'm going to be brief, but I want to tell you one thing, which is, I don't know if you noticed, we're at war. Did everyone know we're at war? We've actually been at war for millennia. We're trying to defeat bugs. Bugs are our enemy. And I've taken a bit of notes from someone who was able to defeat their enemies. Do you know who's... I'm at war inside myself as well. So excuse me for that. Everyone must remember Sun Tzu, the famous Chinese general and military strategist. I bet you read him every night before you go to bed. He says that in order to subdue the enemy, you should try to do it without even fighting. So this is what we do. We blow bugs up. But the interesting thing is that we're looking at a mechanism that all bugs have. It's their survival mechanism. But what if, when bugs are starting to take over, when bacteria are starting to colonize, we can just flip a switch and turn all the lights out? Well, that's what we see when we inhibit specific enzymes in this survival mechanism. So, I'm going to talk to you about this specific switch, this stress response. Busy slide, I know, but I'm a very visual person. I want you to go from left to right, and I'll tell you what the story is. The story is that we have bugs inside us, and sometimes they go wrong. Sometimes they attack us, and we try to get rid of them. We try to treat them, either through our own immune system or through antibiotics. And that triggers the stress response. That triggers a specific group of enzymes, the RSH enzymes. The first one is synthetase. It starts to generate an alarmone, PPP-GPP, and many of you will know this, but that starts the stress response. That's what is causing biofilm. That's what's causing persister cells. That's what's causing virulence. And it then uses another enzyme, hydrolase, to maintain that and make sure it doesn't go haywire. And then when the stress has receded, it'll go back into recovery and recolonate. Now, what we can do is we can block either or both of those enzymes. If you block synthetase, this response doesn't even start. And we can expose the bacteria without these specific defense mechanisms. If we block hydrolase, an interesting thing happens that you've just observed, which is the cells go into complete overload and start to go bang. And this is what we in Centero are developing. We're developing small molecules that inhibit each of these enzymes. What we have so far, and we're at an early stage, we have a lead with hydrolase inhibition, where we're seeing a very strong gram-positive profile. We've got in vivo efficacy with a sepsis model, and that's directly linked to the inhibition of hydrolase. And then in terms of synthetase, we're in the hit to lead phase. We're again seeing very positive, very strong general gram-negative activity with on-target inhibition. And this shows the actual increase in ATP as it's not being allowed to generate PPP-GPP from that ATP. So we have these two mechanisms with one target, this stress response target. You switch it off or you switch it too far up, and these bugs just cannot defend themselves. So the interesting thing is not just that we can look at sepsis models, not just that we can look at being a standard antimicrobial, but we can look at biofilm and persister cells. We've heard a lot this last 2 days about the importance of tackling biofilm, of tackling these cells. It's an incredibly important target for us to resolve. I've got a really nice little video from our founder and his lab. I want you to just follow on with me. I'm going to start it in a second, but you see the big red arrow? Hopefully you do. If you're colorblind, it's the arrow that's red. So it's pointing to one cell. This one cell is going to be the persister cell. What's going to happen is that the bacteria are going to grow, then they're going to be treated, I think it's with ampicillin, with this specific one. This cell will just persist. It won't do anything. And then when ampicillin is taken away, you'll see what happens. So this is what happens. Watch that cell. It's just thinking, it's a normal day, my friends are going away. Oh, it's really nice, I'm going to recolonize. And it does. And that's what we're trying to defeat. So what have we been able to demonstrate on top of the hydrolase and synthetase inhibition, MICs, etc.? Well, we can also show superior efficacy against biofilm and persister cells versus the standard of care, rifampicin. And we also don't see any resistance happening over the long term. We did a very long-term serial passage experiment showing that we just don't see that frequency of resistance. It's transient when it does happen. So we're pretty positive about where we're going. We're still at an early stage, as I mentioned. But with both of these mechanisms and the fact that we have a conserved target across bacteria, no mammalian cell has these enzymes, by the way, so there should be no on-target safety issues. We're looking at two different opportunities. Obviously, we're looking at acute infections. It's something that we are all very, very interested and active in. HAPVAP is the first one. Because of the broad spectrum gram-negative activity we're seeing, we're progressing this. It's a big market. It's not well invested in, and we want to push forward with our technology to really rise to the challenge there. But we also believe that chronic infections are the way forward for our technology. Chronic infections are where the commercial opportunity is. NCFB, for those who don't know, stands for non-cystic fibrosis bronchiectasis. A product last year was approved. It's going to do $1 billion this year, and it was approved last year. It's not even an antimicrobial, it's an anti-inflammatory. And what it does is it reduces the exacerbation rate. If you can do that with both an anti-inflammatory and an anti-infective, then you've got a very, very commercially successful product on your hands. This is what we're going to be looking for in our chronic program. And there's a lot of other chronic diseases that you can see on this slide. But when you put these all together, we have a major opportunity with this one specific master switch that we can turn up or off to create a therapeutic effect. So why am I here today? I'm here because we believe that we need to work as an ecosystem, as a community. We're looking for opportunities to partner. We've got a 15-year head start. That's the amount of academic research and in-house research that's happened in these enzymes from our founders and from the Centera team, who were amazing, by the way. Some of them are here today. We have 0 competitors in the RSH enzyme space. We have multiple targets, and we also have patent protection that covers us for many, many years. So, as I say, we've got gram-negative and gram-positive programs and chronic refractory infections as an additional program. And we're looking to raise money. We're looking to co-invest, get co-investment. And we're just really excited to be a part of the community and to push forward our what I think is an amazing technology. And I look forward to your questions. Thank you very much.
Thank you so much, Mark. Maybe some question in the audience? In the meantime, I'll have one for you. Yes. Is there any preview PK data on your compound? I mean, is there any data on for example, the lung penetration of the compound if you are targeting HAPVAP?
Yeah, so we're in that stage at the moment with our gram-negative program where we're looking at PK. What we found with the series that we progressed with gram-positive was that it had very, very good penetration in the lung. We're hoping we will see that again with our gram-negative program. But it's a small molecule. I don't know if you picked that up. So we're designing it to be able to be delivered IV and oral. We need oral for the chronic indication or some way of delivering it across in the community across multiple months, years, maybe. So PK is a really important thing that we're trying to optimize now.
Thank you.
Yes.
Yeah, one question over there. The microphone is coming.
Thank you. I would like to ask if you observe any resistance happen.
So the only resistance we're seeing is transient, at least with the lead series that we've progressed with the gram-positive side. We're in the middle of doing research into the resistance profile of our gram-negative program. But we think that the resistance is difficult for the bacteria to start generating because we're hitting that very, very early stage cascade where they think, oh, something's wrong. We can't start the response, so we can't start any sort of way of generating a resistant mutation. So it's a really, really important question, especially if you're looking at chronic diseases. But we're seeing so far that there's a very low level of frequency of resistance with very transient resistance when we see it.
Yeah, and I have another question. When you mentioned that it's target the gram-positive and gram-negative, it does the broad spectrum of the gram-positive and the broad spectrum of gram-negative, or is specific to any type of bacteria?
Oh, it's interesting because even though the target is conserved across pretty much all bacteria, we're able to design chemistry that can be more active in certain bugs than others. And we do have series that are very specific around one or two specific bugs, and some that are very, very generally applicable across multiple bugs. In fact, the gram-positive proof of concept I showed, we did test that specific series in a gram-negative infection model as well, and it showed that it was gave a couple of logs reduction in that model as well. So it's something you can see as a broad spectrum antibiotic, but you can also design chemistries to be more targeted. It just depends on the application.
Yeah, thank you very much.
A very, very great pleasure. Thank you.
Yes.
Daniel, maybe?
PK/PD is going to be super important with this.
Absolutely.
The second it's off, it's going to just go, right? So how are you addressing that? I know you mentioned PK and exposure, but when it comes to these different indications, it's going to be interesting, I think, looking at that. So from a dosing standpoint, I guess, and your half-life and all that kind of stuff.
Yeah, I think we're at the very early stage of addressing that. I don't necessarily have anything to say publicly on that. If you've got a deep pocket and an NDA to put in front of me, I'll sign it and tell you more. But we do have a great team that have a lot of experience in designing small molecules in this type of with this type of challenge, and we believe we can resolve that. We've already seen that we can achieve PK enough to get that in vivo model to work with gram-positive with the hydrolase inhibitor. And so it is a really important question. There's other important questions as well that we need to resolve to really get to the point where we know we have a chronic dosing regimen we can achieve orally, or at least in a way that is applicable to patients. But we're rising to the challenge. But thank you, it's a great question. Great.
Okay, thank you, Mark. So
Thank you very much.
I'm afraid I have to close the session. So I would like to thank you again, the four speakers, for this wonderful session. So thank you for coming. And also Noel at the technician and assistants. And good afternoon to everyone. Thank you so much.